Another day, another dollar. Alright, guess I'll just examine the crime scene. Disappointing. Okay. Alright. Using my amazing detective skills, I managed to conclude this is definitely a dead body. Nap time's over, scumbags.
Oh my god, you're hideous!
What? Do you two know why you're here?
No, you kind of just picked us up off the... What is this place?
No, you two are actually under arrest under the charge of murder.
Can you believe this guy? Whoa, bro! Slow down!
Calm down. No need to get really violent.
Son, why are you looking at me?
I'm not.
I said, why are you looking at me?
Oh, God, calm down, bro.
You better find somebody to get us out of here before I...
Whoa! Holy mother of Jesus, who are you?
Sir, I'm the new security guard, sir!
New security guard? What you doing here, boy?
Sir, I was looking for the bathroom on the 3rd floor, sir! When I heard a large screaming, sir!
Well, I never seen you before, so you probably are a new security guard. But you telling me a security guard does not know the layout to a government building?
Sir, education is poor in Africa, sir! I did not learn how to read or write until college, sir!
Alright. Anyways, you might as well help me with the case. Son, I'm gonna be taking some of these guys' individual statements now. Pay close attention. Sir, where were you during the time of the murder?
I was chasing some pretty young girls. They were just walking around, they're dancing, looking. It's very good. It's good entertainment.
Son, you realize that every girl in this building is under the age of 12?
Love has no boundaries.
Anyways, son, where were you during the time of the murder?
What do you think I was doing?
I asked you a question, son.
I was reciting a speech for the KKK for Kids Foundation, aka the KKKK.
Okay.
Okay?
Alright, this interview thing's not gonna work. Son, I think the only way we're gonna get this case done is by using technology.
Technology? Like forensics?
Yeah, please contact the lab boys and ask them what we can do about this case.
Yes, sir. Here's this, sir.
This is a list of all you got? Man, half of this technology is outdated. Not only that, but modern innovations have improved DNA technology throughout the years.
Sir, the last technology we had was a wheel.
So African technology is not that great. Oh well, we'll make do with what we have. Anyways, did any of your boys find any hair samples at the crime scene? If so, we can run a PCR.
PCR? What's that?
Polymerase chain reaction, abbreviated as PCR, is used by lab technicians to create multiple copies of a selected DNA fragment. Here's a generalization of the PCR process. First, to separate the DNA strand and to isolate your target strand, you must heat shock the double helix. Heat shocking is a process by which the DNA is suddenly surged with intense heat. The heat shock effectively removes the hydrogen bonds between parallel strands. The heat shock is nearly at boiling temperature and lasts for about 30 seconds. The next step is the annealing step. In the annealing step, the DNA strand is cooled to about 50 degrees Celsius. This cooling allows the primers to bind with the selected strand. An Taq polymerase attaches to the primer and begins DNA replication in a process called elongation. Fun fact, originally, this Taq polymerase was found in thermophilic bacteria, which is why scientists selected this enzyme for DNA replication, because of its high resistance to heat. And Taq polymerase is an extremely efficient enzyme, creating thousands of base pairs per minute. At this step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template by adding complementary bases to the template in a 5-to-3 direction. As shown in the picture, steps 1 and 2 are repeated again before reaching step 3, elongation. The total process allows for DNA to be replicated at an exponential rate. Finally, the DNA is collected and kept at 14 to 15 degrees Celsius to be preserved for later use.
You have a gel, sir.
A gel? That means electrophoresis. So we can do either the Sanger method or the southern blot.
Wait, sir, what are those, sir?
The Sanger method is used in DNA sequencing. Basically, this method finds the nucleotide sequence of a DNA strand. The Sanger method uses DD-NTP, dideoxide nucleotides, and these DD-NTPs are used to terminate the replication of a DNA sequence. Replication in the Sanger method is done just as in PCR, except a normal polymerase is used and there's no massive amount of DNA product. These DD-NTPs stop DNA replication due to the lack of a hydroxyl group. The termination of a DNA sequence is completely random because the modified dideoxide nucleotides are randomly mixed with the normal nucleotides. All of the strands are then collected and fed into a machine that identifies the type of DD-NTP, and then they are electrophoresised. To find out the identity of a strand, read the electrophoresis gel from down to up.
The process of southern blotting is used in molecular biology to find certain DNA sequence. It's used for homology-based cloning of nucleotides and also used to find methylated sites in some genes. First, restriction endonucleases cut DNA into smaller fragments. Then, the DNA fragments are used in gel electrophoresis to separate them by size. Nitrocellulose is placed on top of the gel, then the membrane is either baked in an oven or exposed to UV rays to attach the DNA to the membrane. The membrane is then exposed to a hybridization probe. A single DNA fragment with a specific sequence whose presence in the target DNA is to be determined. The probe DNA is labeled so that it can be detected, usually by incorporating radioactivity or tagging the molecule with a fluorescent or chromogenic dye. After hybridization, excess probe is washed away from the membrane, typically using an SSC buffer, and then the pattern of hybridization is visualized on an X-ray film by autoradiography.
You know what we actually use for right now? Some riff-lip analysis, also known as DNA fingerprints.
What the doodle do you want to do to the prints?
No, son. Fingerprints.
Oh!
RFLPs, also known as riff-lips, which mean restriction fragment length polymorphism, is basically the process of analysis by electrophoresis separation of restriction fragments. What is gel electrophoresis? Let me explain. There's a permeable gel made up of agarose sugar with a row of wells on one side. Different DNA samples are placed in the wells using a pipette. An electric current is sent through the gel, with the opposite side is positive because the DNA is negatively charged. DNA is then cut using enzymes placed within the DNA. DNA then moves along through the sugar. Since the larger parts of DNA cannot get far through the gel and the smaller parts can, several bands will be formed on the gel. These bands are visible under UV light or able to be seen if the DNA is dyed. These bands can be used to compare genetic similarity to determine the parent/sibling. They may be attached to DNA probes, which are complementary strands that can be detected using machinery. In addition to genetic fingerprinting, RFLP was an important tool in genome mapping, localization of genes for genetic disorders, determination of risk for disease, and paternity testing. There's only one technique left here, and that is the gene chip, also known as the microarray.
Dude, microarrays? That sounds gay.
Hey! You can't make fun of a person for his sexuality. They're normal people like us.
But you can make fun of the rays. Yeah.
So, am I going to explain the microarray or what?
Microarray allows for the analysis of hundreds of genes at once. At first, a fragmented DNA is labeled with a fluorescent dye. Then it is placed into a microarray. A microarray is a small chip containing cDNA, complementary DNA. The microarray has many different types of complementary DNA strands to these genes. Each fragmented gene matches up with the complementary DNA strand in the microarray. Finally, the microarray is scanned to show an array of colors, and each color identifies the type of gene.
That's it. I know who the killer is. It's you.
Me? What?
That's right. It's obvious that you were chasing ladies around the building. And guess what? There's only one lady in here. The king's daughter. It's obvious that while you were chasing her, you had an encounter with the king. And so you had to kill him. Otherwise he would execute you for being a pedophile.
Oh, well, anyways, in this country, there's only one way you can arrest me. And that's if we have a sing-off.
People with their boyfriend and never let you go. I get to new places you never been before. Flag, 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 flag on you. Flag, 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 flag on you.
Let's get down to business to get the case done. Got to study for bio, must get home by 1. PCR's polymerase sticks to the primer. After heating, splits it into a copy DNA straight from you. With flip analysis, enzymes cut fragments used in genome mapping, gene profiling.
You're pathetic.
Pressing lock, and you haven't got a clue.
I'll just cut DNA out of you.
Never gonna make it home for lunch, but if my parents love me, going to see the new episode of Him Yim. All non-watchers receive death. What if I can stick them a roofie? It's like wonder that you make me gay.
Sanger method reveals nucleotide sequence. We bottom to top using the gel. Southern blot test for the mutations. This stupid case really must be finished soon. Time is racing toward us before AP tests arrive. Study hard for bio, you might get a 5. I soup forks, I'm that hardcore. You misfits are nothing new. How could you idiots kill a dude? To succeed in bio requires patience. You must understand and study till noon. The culprit is what I must acquire. Vicious as the dark side of the moon. To succeed in bio requires patience. You must understand and study till noon. The culprit is what I must acquire. Suspicious as the dark side of the moon.
You know what? I think Taha's a better singer. Method.
That's it. I know who the killer is.
It's definitely you.
Son, you're free to go.
What?
That's right, son. Just think about it. You hate Jews, you hate Democrats, you hate anyone that ain't a Nazi. You're a non-white, libertarian scumbag. What better way to show your disdain for Africans than to kill the lead African himself, the king?
Well, good job showing about my ideologies. You have no hard evidence that I'm the killer. Who are you?
I don't need hard evidence. This is Africa. There's only one way to settle crimes here.
No. Stop! Stop! Stop!
Whoa! It was only a dream. We've been watching you.
I was asleep. What am I doing in a bathtub? But more importantly, I was asleep. No way, it was all a dream.
guess it was only just a dream.
Alright, so anyway, did your shirt just change color?
Oh my goodness.
No! But the world around me does.
Anyways, I contacted the boys at the lab, and apparently we've found some hair at the crime scene. It's only a matter of time until we carry out a polymerase chain reaction and copy the DNA enough until we find out who the true killer is. So just beware, boys. PCR will truly reveal the...
Oh my goodness gracious, he's got a gun!
I'm afraid you can't do that, sir. Just drop the gun and put your hands behind your head.
No need to get hasty here.
Alright, Mr. Detective. Wait, is that a DS?
Well, I learned how to untie handcuffs back in neo-Nazi camp.
How'd you even bring... Okay, my main question is, how'd you bring the DS in here?
Through legal means.
Dude,
I'm a racist.
just like, you know,
What's
just put
wrong?
that in your pocket.
This is extremely...
Just be glad we're not the killers, so you know, just...
We're having
put
a
it away,
moment
sit
here.
back and enjoy the show.
Okay, jeez.
Oh, your parents.
I know.
Kids in there.
You're a
are-
disgrace,
You know, I think we need discipline.
man.
I agree, I completely agree with you.
Can we just move on,
Okay,
guys?
so, what do you think?
What do you mean, what do I think? I think kids are really shameful today with their electronics.
I agree. But what do you think about who killed the king?
You're telling me you're the killer?
Not just the killer. I'm the prince.
What? The prince? Why would the prince kill his own father?
And I'm not going to get to that yet. But I do have a test for you. You think you're so smart, Mr. Detective, with your stupid math. But I'm about to test that. Let's play a game. Well, Detective. We're going to play a game. It's going to consist of 7 questions. You get them all right, which is really unlikely, you're free to go. But if you get one wrong, I'll make sure you don't see the next iCarly episode. Got it?
But what I need to know if Sam ends up with Freddy.
Dude, can you shut up? God.
Wait, was that one of your questions?
Well, here's that as if it was. Okay.
Okay, but I really need to see the next iCarly episode because I didn't know if Sam ends up with Freddy or Carly ends up with Freddy.
Why are you so annoying?
Was that another one of your questions?
Well,
Looks like you only have 5 questions left.
I'll play it. Okay. First, real question out of my 5 questions that are left. What kind of charged probes are used at the end of the electrophoresis gel opposite of the wells?
Positive, because the phosphate group of DNA is negative.
Good job. What does RFLP stand for?
Restriction fragment length polymorphism, sir.
Not bad. Not bad. How much do you hate Siddharth Mehta?
I hate him a lot.
That was an easy one. Well, that was an easy one.
Yeah, everyone hates him.
Okay. In the Sanger method, how are restriction fragment of DNAs read?
From bottom to top, sir.
Not bad. Not bad. What are microarrays?
Microarrays are used to analyze hundreds of genes at once.
Crap. Alright. Like I said, I'll keep my promise. I'll let you live. But just as a warning, a bomb is about to explode here in 10 minutes.
Come on, she's after him, boy. Come on, help me out. You have a gun?
Yeah.
Oh, okay.
What about him?
You can just stay here. Let's go.
Let's go.
Wait. There's a reason I did this. My father beat me every day at chess. I had to put an end to it.
Listen, I feel your pain, but that doesn't mean you can kill a man. We have to end this chain of killing right now by killing you.
Okay, come on. At least give me a 10-second head start. Like
Okay,
I said.
Yeah, sure.
I mean, he waited first. We might as well give him a 10 seconds.
Alright, okay, so
Okay.
I'm gonna start, okay?
Alright, so sure.
Okay,
10, 9,
8, 1!
Oh my. Sir, you know what? This isn't working. So you know what? I have a good idea. I play Call of Duty a lot, so maybe I should go. I can handle this. Don't worry.
Alright. But just in case you don't make it, Give my hat back. Oh my god, oh my god, oh my god!
I got it! Form his hat! Oh.
No. That suspect was my favorite acquaintance. That's it. Time for some southern revenge. Stop right there.
How did I miss all those?
Looks like you can't electrophoresist the law.
That was so bad. So literally bad. I've lost all faith in humanity. I'm gonna kill myself.
No, don't do it, don't do it, don't do it! Oh well. No! Oh, there's my head.
Come here. Tell my father.
I will.
Swag.